I'm Sara Sunshine, and today I'm going to be talking to you about bioinformatics and how you can use JavaScript tools to explore your genetic sequence. So we have a lot of DNA. The human genome is about 3.3 billion base pairs long. And back in the mid-90s, when the federal government undertook the massive endeavor of sequencing the full human genome, it took 13 years and cost $2.7 million. And at the time, scientists working on this project thought, this is great, and we're really glad we're doing this, but it would be wonderful if we could just sequence a full genome in a day for just $1,000. And the idea of the $1,000 genome emerged as the price point at which consumers would actually go out and get their genome sequenced, use that information in their medical treatment, and also the point at which scientists could afford to sequence large numbers of genomes in order to sort of tease out some of the subtle associations between mutation and disease. And so since the Human Genome Project, the cost of sequencing has plummeted and outpaces Moore's Law. So you can see in January 2008, that's when things really accelerated as next-generation sequencing techniques started entering the lab. And nowadays, we're more or less at that $1,000 genome point. So no longer is getting the data the problem, but the bottleneck is now understanding and interpreting your genetic information. After all, getting your sequence for only $1,000 is not a very good deal if you can't do anything with that information. That's just sort of like an expensive flash drive. So this is where the field of bioinformatics really comes into play, leveraging the power of computing and analysis to extract meaningful insight from our genetic information. So bioinformatics is a massive field, but I'm going to focus on some of the things that have come out of genome-wide association studies, which are large studies looking at many different genomes to try and identify small mutations that are associated with traits or disease. And what genome-wide association studies are looking for are just single base pair changes or single nucleotide polymorphisms, which are called SNPs. And that is, as the name implies, just places in the genome where there has been a single base pair change. Perhaps most people in the population have an adenine or A at a position, but then a small subset of people might have thymine or T, so that's a SNP. So I'm going to walk us through a small example of how to use JavaScript to explore your genetic sequence. So I have not actually gotten my own genome sequenced. This sequence comes off GitHub from one of the brave souls who feels inclined to publish their genetic information for the world to see. And you can see here that there's the RSID, which is a unique identifier for a SNP, as well as information on the chromosome, where on the chromosome it is, and what this person's genotype is at that chromosome. So a reminder from high school biology, you have 2 copies of every chromosome, one from your mother and one from your father. So you have a combination of 2 base pairs at each position. And so this file is 1 million lines long, which makes the Webpack bundle.js files pretty manageable in comparison. So as I said, 1 million lines long, not 3 billion, which is how many base pairs we have. 23andMe is not sequencing your full genome. They're looking just at a predefined subset of SNPs that are known to be associated with disease or some interesting traits. And so this file needs to be converted into a giant JSON object for some of the work we're going to do in a second. So there is a library that can take these text files and make them a JSON object where the key is the RSID identifier. But I was having a little bit of trouble with it, so I decided to implement my own version using streams and Node. So I'm going to take a second to talk just quickly about streams. I think they're a really cool, exciting tool. So as you know, Node is a single-threaded environment, which means that all I/O operations need to happen in an asynchronous manner. And this can be tricky because it can lead to sort of a callback pyramid of doom, or you may have to write a massive amount of data to disk and then read it off. And streams allow you to sort of circumvent this in a really efficient way. So there are 3 different varieties of streams. First, there are readable streams, which, as the name implies, are sort of reading in the data. In this analogy, it's a sort of faucet that you're turning on. And this is how you initialize them in the Stream API. There's also writable, which consume the data. And then, finally, there are streams that can take both an input and an output, or even perform some operation on the data. And those are duplex and transform streams. And where streams really become powerful is when combined with pipes, which you may remember from the Unix workshop. So this is just sort of a simple example of you're creating a readable stream from one file, and then you were just piping that directly into a second file's writable stream. So when I wanted to convert the sequence into JSON using streams, I first created a readable stream from the text file, and then I piped it into a transform stream called the split2 library. So streams chunk up data in different ways, and I wanted it to be broken up line by line. So the split2 library handles that for you. And then I pass that into a transform stream, which filters out comments, and then pipe that into yet another transform stream, which split up the lines into arrays. And then finally, so streams are event emitters as well, and they will emit a data event each time a chunk has been processed, or a line in this case. And then on the data event, I sort of stored the line in an array, and then once the end event was emitted, because the stream was finished, I ran a transformation and reduced this into the giant JSON object. So you'll notice at the end I'm losing some of the advantage of streams because I am writing this all to memory, and I'm performing a synchronous operation. But because it had to be in this JSON format, that was a bit unavoidable. And we can move past this. That's just my transform and reduce operations. OK, so decades of research has allowed 23andMe to just send you a text file with your DNA sequence. And then we can use streams to convert that into a JSON. And once you have the JSON, we can turn to SNPedia to help understand what we're looking at. So SNPedia is this massive online wiki that catalogs all the different SNPs, their associations to disease or drug resistance, et cetera. And they format this information in what's called a genoset, which is just a Boolean criteria for whether or not you have a certain genotype that it will evaluate to true or false. So for a simple example, this is a genoset for if you're immune to neurovirus, which is that nasty virus people get on cruise ships that causes intense gastrointestinal distress, which is probably not fun when you're out at sea. And so the genoset is just at this position, does this person have AA? If so, then it's true and they are immune to neurovirus. And so that's a simple example. They can get increasingly more complex depending on what the literature says. Here's a slightly more complicated one. If you have any one of these 4 genotypes at the respective position, if you're of European background, you are 77% likely to be able to digest lactose. So these kind of criteria, the genosets, they're built using semantic search, which I thought was really cool. So in essence, that means that these RSID numbers are unique. They're used in every database and every publication that refers to this SNP. And so SNPedia is run by just basically a bot that crawls literature, and anytime a paper like this is published, it will notice the tag for the RSID, pull it, and use that information to compile the criteria that's contained in the genoset. And so we can couple these genosets with JavaScript's genome query language, or GQL. And this is a package that was developed by Eric Schafstall, who's one of the engineers behind Gulp.js. And in essence, it is much like SQL, will produce a query function that will look at the database of your DNA and return whether or not you can find the genoset in the DNA. And so these genosets, I should add, they're just available as NPM packages. You can install them. And here's one for male pattern baldness. So GQL is just a really lightweight library that's sort of a natural extension of the logic you would expect from a query language. And if you look under the hood, they're just doing some sort of functional programming to generate these query functions that you can then pass your DNA into. OK, so we got the sequence from 23andMe, converted it into JSON, and now we have a query language that we can actually use to evaluate whether or not the SNPs are present in your sequence. So these are some of the more common SNPs on SNPedia that people look at. You see these are a little more serious in nature, particularly genoset 216, which means you have 2 copies of the ApoE4 mutation that's associated with early onset Alzheimer's. But there's also ones that are a little bit more fun and lighthearted, like if you are more stimulated by coffee than most people, or if you have a predisposition towards red hair, which I'm not sure why you would need to be assaying your genome to determine that. Hopefully you could just look in a mirror. And my personal favorite is genoset 302, which means you have facial age perceived as 1 year older than average. Which brings me to my final point. You can see that some of these are getting a little hand-wavy. So I sort of have a few caveats. I'm really excited about genetics and bioinformatics and would encourage everyone to kind of look into your genome. But I think the concept of relative risk is something that gets lost in these discussions sometimes. So if you have like a 3% increased risk of something that's very rare to begin with, there's no need to sort of panic and lose your head about it. Furthermore, bioinformatics is a really young field. We're constantly learning new things. So more information might become available. And along with that, with 3 billion base pairs that are being looked at for any possible correlation, you're going to have some things emerge that seem like there's a really strong statistical connection, but in fact, it's just what we call a spurious association, like the number of people who drown by falling into a pool and films Nicolas Cage has appeared in. It seems like there is a connection there, but probably not. Although, who knows? And then finally, if you choose to do this, I would encourage you, for anything that you find, to look at the source. What's the quality of the evidence behind it? Is it just 1 paper, or is it 5 papers? Is it from a reputable journal like Nature or Science, or is it from maybe scientists who aren't as reputable? And then finally, if you do find anything that gives you cause for concern, I would really urge you to consult a medical professional, as I am not a physician. I don't think any of you, except for David, in this room are. So one of the criticisms of 23andMe and SNPedia is that this kind of information is supposed to be processed in the context of a genetic counselor. And I do think there's some validity to that. So if you're interested in learning more, here are some resources. And I'm happy to talk afterwards. My slides and code can also be found on my GitHub. And thank you. You've been great.